Many thanks for your kind introduction, and many thanks for the Jung Foundation for this incredible honor to speak here, and also the award. It's really an amazing experience. So I would like to share some research that our group is doing on bacteria. And of course, some bacteria are the bad guys that cause diseases, but many more bacteria are actually beneficial for humans. And one reason is that many bacteria produce bioactive substances that can be used for a wide range of therapeutics, including most classes of clinical antibiotics that are available today are based on either the actual natural product itself discovered in a bacterium or on a pharmacological principle, a part of a structure where it was found if it's incorporated, for example, in a synthetic molecule, then it's a useful antibiotic. So when we take a look at the course of natural product discovery, so this is for general natural product discovery, also from other sources, then we see a trend that might look a bit concerning because the overall trend is going up. So the rate of natural product discovery, we have more and more natural products. But if you take a closer look, then these are more or less variants of already known compounds. And this curve here shows distinct structures. So you can calculate this with a Tanimoto index. This was published in this paper here by Roger Linnington and co-workers. So this might look concerning because it might suggest the pipeline is running dry. And what is the message of my talk? To hopefully convince you that this is actually not the case. On the contrary, that we might have barely scratched the surface so far. And I would like to illustrate this by showing you this very complex bacterial tree of life published by Jill Benfield and co-workers. You see these tiny labels. I don't ask you to read all this, but just the impression is there's a lot of diversity. And these labels refer to phyla. So these are very deep-rooting, very ancient taxonomic units in the plant world. Flowering plants is, for example, a phylum. And you see there are lots of phyla. But if we now map the available sources or the studied sources of bacterial natural products onto this, so these are these yellow dots, then you can see that this is quite sparsely covered. And what these are mostly are so-called talented producer groups. So these are bacteria where one strain produces often not one bioactive natural product, but a whole battery of them. And one famous talented producer group are these soil-derived filamentous actinomycetes that have provided many bacterial-derived drugs. It's convenient to study these talented producer groups in these traditional screens because you go ahead, collect or study randomly bacteria, extract them, and then test whether the extracts have a bioactivity, and later isolate the corresponding compound. So you would have high hit rates in these initial screens. But if you look at these desert areas, as it were, and you look into the genomes, then you would see natural product biosynthetic genes in almost any of these bacteria. It's just much more tedious to study them. And another reason, and I think a much more important reason, is for why we have barely scratched the surface. It lies in these red dots. What these signify are phyla, or in this case you call them candidate phyla, that do not have a single cultivated representative. And you see this amazing number of basically unstudied bacterial life. And even more, if you would zoom into these groups here without a red dot, you will see at the subphylum level many more of these groups. So it's quite surprising how little we actually know about bacteria. And so we have genome sequences or partial genome sequences, but we know very little in terms of function. And when I did my postdoc, it was 1998, we didn't have this very detailed resolution of the bacterial tree of life, But for some years, the concept of the uncultivated bacterial majority was known. And then in 98, I encountered this seminal opinion paper, which stated that this was for soil, that this uncultivated majority might be a new source of, an untapped new source of new chemical entities that you could use for drug discovery. And this was published by Joe Handelsman and other giants in the field, and it was actually the first, to my knowledge, the first paper that used the word metagenomics, so the study of whole collections of genomes from a community that is widely used today. So I found this extremely fascinating. It triggered my entire later research career. So the questions that we are looking at, and I will address them individually, is there actually new chemistry? Do we even have talent producer groups? And the most important question, of course, if you're just starting a group, what kind of models are suitable for this? It sounded a bit scary and challenging, and what kind of methods would you use? So I will first address the last question. As I mentioned, this paper advertised soil, but we liked another type of system much more, which is host-associated microbiomes. And the reason was that these were almost like bioreactors that could grow uncultured bacteria that you cannot grow, and they are spatially defined. You can recollect them, and the microbiomes would be, we hoped, stable, but that was actually not necessarily the case. And another reason we thought would be interesting is that the ecology would be much different from the soil bacteria, of which some groups were very well studied. So we thought this might correlate with new chemistry that has been selected in evolution. So if you want to study biosynthetic genes, I would like to show you quickly how these are organized in bacteria, because this is something very convenient to study. In bacteria, usually all the genes that you need to produce one particular compound are clustered together. So these are biosynthetic gene clusters, and many of these genes encode enzymes, which have a particular function in assembling the metabolite. For example, this would be a methyltransferase that attaches a methyl group, and so on. So if you find maybe one of these genes, then you would know all the remaining genes are left and right on the genome, and you have a functional package that you even might hope to transfer into cultural bacteria and then produce the compound. So how do you find these gene clusters? These days you can just sequence everything by metagenomic sequencing, but that wasn't possible at that time. And the method that was also proposed in the paper, it was transferring pieces of this foreign DNA, this metagenomic DNA, into a host such as E. coli to create libraries, and then you would screen these libraries for based on some screening method, for example, PCR, based on a biosynthetic hypothesis. And then this, of course, this is just in silico prediction, then you would have to follow this up by characterizing enzymes, for example. These days it's much more easy, at least up to here. You can just sequence everything by metagenomic sequencing, or there's even single-cell genomics where you can amplify the DNA from just one cell and sequence it. So I should mention the collaborators to whom I'm really grateful. I cannot list all of them, but here are the most important ones, 3 groups in Japan and one at our institute, Shini Sunagawa. And together with these people, we had a lot of fun after we overcame the first hurdles with playing with these environments. So this was a bit of background, and now I would like to show you what we found. And the first topic will be the producers, so what kind of producers did we identify, and what are their compounds? And then the question is, if you cannot cultivate a bacterium, how do you get hold of the compounds? And that will be part of the second section of my talk. So let's first start with producers and the model systems that we worked with. I showed a bunch of hosts, sponges, insects, and so on. But the organisms we were most fascinated by were marine sponges. And the reason is that they are two treasure troves of bioactive compounds. So one of the richest known source for drug discovery as well. And there are some drugs available, not many of them. This is one, and this inspired the concept of nucleoside analogs, which is actually a larger class of bioactive compounds used in various drugs. And this is used, this sponge compound, in an anti-cancer drug, but also remdesivir, this coronavirus drug, is based on this initial discovery in sponges. So these structures are quite simple. They are accessible by chemical synthesis, but this is unfortunately not that easy for most of the other sponge compounds. And I show you some drug candidates and also another compound. This is a commercial cell biology tool, but you see this is pretty unsustainable. It's sourced from sponges, and the amounts you get are not very high. And here are some very attractive drug candidates, anti-cancer drug candidates. Pyloroside has a new binding site at microtubules, and this is a new mode of action for boxazole, and it's an extremely active compound. And the supply is very limited, and to address this, in this particular case for pyloroside, New Zealand groups have tried to establish maricultures, but these were invaded by a snail that ate the sponges to sequester the toxin for their own chemical defense. So that was the end of this experiment. So I mentioned if you are, first I should say, why am I now talking about sponge compounds? I started with bacteria. So if you look at these structures here with a biosynthetic eye, then they look very suspiciously like complex polyketides. And this is a compound class that is very common in bacteria. And that's why it has been proposed for a long time that these are actually produced by bacteria residing in the sponges, in the sponge microbiome. And for us, we thought, if we would find a biosynthetic gene cluster from a bacterium, for example, for boxazole, this would be amazing. But at the same time, we thought this is a doomed project because very little was known about the sponge. We didn't know how many bacteria, what else, what other natural products are in there, and so on. So remember, at that time, of the three methods, we only had this one available. And for every gene cluster, you would have to invest in new screening efforts, screening millions of clones. This is really painful. So we thought to be more realistic, to start with something else that is maybe not a sexy drug candidate, but can give us faster insights into producers and sponges. And to illustrate this complexity challenge a little bit more, here is Thionella swinhoae, a marine sponge, containing a whole suite of compounds with various bioactivities. And the sponge that was a bit better studied at that time, and it was known it harbors about 1,000 different bacteria. These compounds look really strange, for example, this one here. And also you have chemical variability, so depending on which specimen you have, compounds might be there or not. But still, we thought this is a great model system because there was one compound here that might give us access to producers. And this is because there was another compound known that was almost identical, but from a rather unexpected source, which is terrestrial beetles. And these look quite colorful, but I don't recommend to play with them because this happens when pederine, this compound, comes into contact with your skin. They use this as a contact poison for chemical defense. So these beetles had a much simpler, smaller microbiome, and we thought it would be much easier to find the biosynthetic genes here and the producer, and then maybe by gene comparison would be able to find similar genes in the sponge, and then from there know what a producer and a sponge would look like. And this worked really well, to our surprise actually, and shown here is the pedigrine producer, and it's indeed a bacterium, and this is also a bacterium, but it's a completely different bacterium. And it was so exotic that we proposed the existence of a new phylum or candidate phylum that we called Tectomicrobia. But these filaments were known before and proposed by Margot Haygood, Eric Schmidt, and Carol Bruley, and John Faulkner, to be a producer of another sponge natural product. So now that we knew that these are large filamentous and can be mechanically basically enriched, we were also able to sequence these filaments. And then our surprise was even bigger, because the genomic data suggested that this bacterium, among 1,000 bacteria in the microbiome, produced not only onomide, but almost all of the other compounds in the sponge, except for this antifungal metabolite here. And we found even more biosynthetic gene clusters for as yet unknown compounds. So this was the first talented producer from microbial dark matter. And it was even a talented producer taxon, because then, of course, intrigued by this finding, we looked at many more sponges and checked whether we could find these filaments in there. And whenever there were filaments, we could sequence them, and we've got a very consistent picture. Every time these endothionella filaments were present, we found on the genome gene clusters for either all or almost all natural products. So here I would like to show you some very busy slides summarizing the information on endothionella, so various endothionella candidate species. So this is a very diverse taxon with a very variable metabolism. And I should mention this was not our work, but the main work of our collaborator Toshi Wakimoto, and we just helped a little bit with interpreting gene sequences. This is the commercial cell biology tool, by the way. So now you might have the impression endothionella generates every single natural product found in a sponge, but that's certainly not the case. There are many sponges that are chemically rich without harboring these filaments. And here we studied some more. This one, quite opposite to the endothionella situation, this sponge contains a small microbiome where almost every member contributes one or two compounds. And to our, we were very glad to see that we also were able to isolate the palerocyte biosynthetic genes, so for these failed mariculture attempts, and identify the producer. And this is the remaining compound in the initial sponge that was not made by endothionella, and this is another producer type. So because this was a lot of information, I give you some take-home messages. And one is microbial dark matter indeed contains producers with an actinomycete-like chemical richness, but the compounds are very different from what you find in these typical industrial producers. The producers are also very exotic, so every time we look for a source of a known compound in a host, then we have to give it a new name. So these are all names given by us, and often not only for the candidate species or genus, but higher order taxa, families and orders, and in one case, as I mentioned, also a phylum. So a lot of phylogenetic novelty, and this correlates also with biosynthetic novelty, which I would like to explain on the next two slides. So I would like to show you two new natural product families that we discovered in this context. And one concerns these complex polyketides in sponges that I mentioned. Polyketides were a known compound class also from actinomycetes, and the biosynthetic enzymes were quite well studied, the polyketide synthesis, and that's why we were so surprised. It's in the beetle we found them first, to see that these ones are made by a new family of polyketide synthesis that we called trans-AT polyketide synthesis. I won't explain why. It's just a new way how complex polyketides are synthesized. And These were overlooked because they are not that common in actinomycetes, but they are quite common in lesser studied bacterial groups. And for some reason that we still don't understand, they are predominant in bacterial symbionts from various hosts. So we haven't found these genes, hadn't found, but we predicted that this would also be made by such an enzymatic system. And also by targeted genome mining, so genome-inspired natural product discovery, we could study some of these other bacteria, and the discovered natural products had a wide range of bioactivities and also potent activities, some of them. This was one of the natural product families that were new in these organisms, and this is a member of another new one, a crazy structure, extremely complex peptides, and it turned out that these are post-translationally modified peptides where all these colored regions are post-translational modifications, very strange ones. These blue regions refer to D amino acids. It's one enzyme that installs 18 D amino acids. These are polythionomides, previously discovered from the sponge by Shigeki Matsunaga, and these are extremely potent compounds, activity down to the lower picomolar range. These are good candidates for antibody drug candidates. And these were the first members of a new peptide family, actually, that we called proteosins. And they are characterized by a very rich suite of novel post-translational modifications, also in addition to these ones. And also the activities that we identified are very diverse. All right, so this was about producers and compounds. And now maybe you ask yourself, these bacteria still cannot be cultivated. How do we get hold of these compounds? And one tempting approach was when we looked into the genomes, sequenced, for example, the Anthocyanella genome. They looked very large and complete. Not much was missing, and we thought, yeah, let's just try and cultivate them. This was really frustrating. All trials failed for many years, not only by us, but also by several collaborators. Extremely challenging, so we gave up. It's also frustrating for PhD students, for example, to do the same experiment all over again without any hypothesis. The other strategy I mentioned briefly, which is if you are able to identify the biosynthetic gene clusters, you might be also able to transfer them into cultural bacterium, E. coli, for example, and then produce the compounds. And this worked for example, polythionomides and related metabolites. So we have now production systems where to create a new host for this because some of these modifications are challenging for E. coli. And that also allowed us to generate analogs, natural products as it were, including protein conjugates. And now a heavy focus in our group is, since we have these pylorocyte genes, to create a sustainable production system for this compound as well. This looks much easier because it's such a simple compound compared to polythionomides. But if you look at the genes, these gene clusters are gigantic. These are small here, paradoxically. And this is challenging. It's a multi-year effort for every single compound that you want to produce. But yeah, we think this might work someday, but for a routine strategy, large-scale strategy for a lot of polyketides, this is not really suitable for a single lab. And that made us think, is there maybe a third alternative strategy to get hold of particularly these challenging polyketides? And the idea was, can you predict from bacterial genomes, genomes of bacteria that can be cultivated, if these produce the same compounds that you are interested in? And I mentioned that these are generated by novel, unusual enzymes, and these look horrific. If I show you the architecture of an enzyme on the next slide, it's not that straightforward either. But the rationale why we thought this would work is as you have seen for this beetle and the sponge case, sometimes biosynthetic gene clusters are horizontally gene transferred even between very distantly related bacteria. We thought we might be lucky and identify a culturable bacterium with the same gene cluster. So the goal was, can we computationally predict such compounds from genomes? And ideally, even, for example, for boxazoles, compounds that we don't have any biosynthetic genes for, because the sponge was not accessible. But a prerequisite is if this is a new enzyme family, is that we have to understand how they work so we can correlate the genes with natural product structures. This is how a typical trans-AT polynucleotide synthase looks like. It's a total nightmare. And when we found the first example, we had no real clue how do these enzymes work. All these abbreviations refer to different components that have a different catalytic function, and many of them are fused to huge enzymes, and we also have non-covalently associated components, a total mess. And we spent many years to understand how these enzymes work, and also other groups were interested in that and contributed to an understanding of so-called biosynthetic modules, because this is the principle of these enzymes. And I can show you a simplified version of this, where you just see the modules, and each color corresponds to a different function of a module that installs a different chemical moiety in the chain. So, for example, here you have a ring, and there's a module that installs a ring, and this is followed downstream by a chlorine atom, and here is a module that installs a chlorine atom. So after we got all these insights into module functions, it was then even possible to predict fairly precisely how, except these macrocyclic ring closures, how compounds would look like that we predict from genomes. And the goal was then to see if we can predict a bacterium that would produce a sponge-type metabolite. So in order to achieve this, we collected all the genome information from public databases, and what you now see are these tiny radial lines, and each one, this is actually the output of the computational tool. It looks like this, and it tells you what the compound would be, and we have another tool that translates this to the chemical structure. We thought we had to tediously predict all of these more than 5,000 polynucleotide synthase products, but to our surprise, we just played a little bit with it, and we found already a very interesting producer group, and for exactly this type of compound, which was unbelievable luck, I think. So these 4 boxazoles. And caliculin as well, this commercial cell biology tool. What are these bacteria? These are also marine host associated. Many live in corals, but also mollusks. But in corals, they are most commonly found. Many of them are uncultivated, but these 2 can be cultured. And of course, we ordered them. This was predicted to produce both types of metabolites. This one, only one, but this was more similar to almost identical to the actual natural product that was first known. So we grew up both, and we could indeed isolate the compounds and characterize them by NMR. So now we have a renewable source for these metabolites. What are these bacteria? These are members of acidobacteria, a very broadly distributed, also in soil phylum, but only a few members are cultivated, but they are apparently culturable with the right tricks. And here, for example, this one, in addition to a biosynthetic gene cluster for this compound, there are 45 other biosynthetic gene clusters, and this one has 30-something more. So this is another talented producer group that hasn't been chemically studied so far. And with Shinizu Nagawa, you now look at coral microbiomes, and again, you see this incredible richness of biosynthetic gene clusters. So I think this is a very promising group to focus cultivation efforts on. So this is the end of my talk. I'd like to quickly summarize. I hope I could convince you that uncultivated bacteria are still largely untapped. This is just little glimpses of it, what we had. Untapped resource of novel producers, but also new natural product families, new types of enzymes. We think it's very exciting. And many, but not all, sponge natural products are made by bacteria. I didn't mention it, but Eric Schmidt's group, for example, showed that some sponge peptides are made by the sponge itself, and there are also many other bacterial symbionts. And then how to get sustainable access. I explained heterologous expression, then finding related pathways. The more bacterial genomes get sequenced, the higher the likelihood that you find these alternative sources. And maybe, I'm sure someone will succeed in cultivating some of these organisms. Yeah, in the end, I would like to thank a lot of people. Unfortunately, I couldn't show pictures of all of the fantastic people involved in these projects. I'm really, it's amazing to work with these very talented scientists, and also lucky to have a lot of great collaborators. And of course, I would like to thank funding agencies, especially the Jung Foundation, for this prestigious award. Thanks a lot for your attention.